Phase 2 is the inflection point that defines portfolio economics. Phase 1 establishes safety. Phase 3 confirms scale. But Phase 2 determines whether biological promise translates into measurable clinical benefit. When Phase 2 fails, the impact is not only financial, it reverberates across the portfolio. Years of discovery, preclinical validation, and capital allocation converge at this stage. The strategic objective of translational medicine is clear: reduce uncertainty. Before Phase 2 begins, artificial intelligence, particularly machine learning, provides a direct path toward that objective. Disease is rarely uniform. Patients who share the same diagnosis often exhibit different molecular drivers, pathway activations, immune responses, and disease trajectories. Traditional clinical trial design treats populations as broadly homogeneous with predefined inclusion criteria. Machine learning changes that assumption. By integrating genomic, proteomic, transcriptomic imaging, and clinical phenotype data, ML models detect biologically distinct subpopulations. Instead of enrolling broad, heterogeneous cohorts, organizations can identify patients with a higher molecular likelihood of response. This is machine learning-driven stratification. Supervised learning models trained on historical responder and non-responder data reveal combinations of biomarkers associated with therapeutic sensitivity. Rather than isolating single predictors, ML uncovers multidimensional response signatures. Uncertainty is not eliminated, but it becomes measurable. If enrollment includes patients with low predicted response probability, statistical failure becomes more likely. If enrollment is enriched with high probability responders, signal detection strengthens and power improves. Phase 2 becomes more controlled. The goal is alignment between mechanism of action and patient biology. Translational medicine becomes computationally augmented. Digital biomarkers expand this leverage. Traditional biomarkers rely on static laboratory measures. Digital biomarkers derived from wearable devices, imaging analytics, continuous monitoring, and behavioral tracking capture dynamic disease evolution. Machine learning analyzes longitudinal streams to detect subtle shifts in disease state or therapeutic effect that conventional endpoints may miss. In neurological conditions, digital motion tracking may reveal functional improvement earlier than clinical rating scales. In cardiovascular disease, continuous rhythm monitoring may indicate stabilization before overt events occur. These richer signals provide earlier clarity. For leadership, this translates into faster and more confident decision-making. If early data indicates limited physiological change, strategic termination can occur before capital escalation. If early stratified signals are strong, Phase 3 investment gains stronger confidence. Economic leverage improves. Deep learning enhances imaging analytics. In oncology, radiomic models analyze tumor heterogeneity beyond visual inspection. These subtle features correlate with treatment response and disease progression. When imaging-derived biomarkers integrate with genomic and clinical variables, multimodal predictive frameworks emerge. Responder modeling becomes more precise. Predictive responder modeling assigns probability scores to individual patients before enrollment. Instead of observing response after exposure, organizations estimate likelihood in advance. Trial design shifts from broad recruitment to targeted enrichment effect size. Within enriched cohorts, increases, statistical power improves, sample size requirements may decrease. Regulatory agencies increasingly recognize precision approaches when supported by validated evidence. Validation discipline remains essential. Retrospective models must be tested on independent cohorts. Overfitting must be avoided. Generalizability must be demonstrated. Governance preserves credibility. Another strategic dimension involves mechanism alignment. Machine learning can evaluate pathway activation patterns in patient samples. If a therapy targets a specific signaling cascade, ML can identify patients in whom that pathway is active. This increases mechanistic coherence. Without alignment, a scientifically sound molecule may appear ineffective because enrolled patients do not exhibit the relevant biological pathway. Failure in such cases reflects selection, mismatch, not scientific invalidity. AI reduces that mismatch. Digital biomarkers also strengthen safety forecasting. Continuous physiological monitoring analyzed through ML can surface early warning signals before severe adverse events occur. Patient protection improves, regulatory exposure decreases. For CROs, stratification analytics and digital biomarker integration enhance competitive positioning. Sponsors increasingly expect data-informed enrollment strategies and adaptive oversight. For sponsors, embedding predictive modeling into translational planning improves portfolio discipline. Not every therapeutic area requires deep stratification. Oncology, immunology, neurology, and rare genetic conditions often benefit most from enrichment strategies. Broader metabolic or cardiovascular indications may require alternative modeling approaches. Strategic discernment is critical. Adaptive trial frameworks further enhance agility. If early interim data reveals differential subgroup response, enrollment criteria can be adjusted within regulatory boundaries. Machine learning supports continuous probability updating as new data accumulates. Time to insight shortens. Transparency remains essential. Regulatory discussions require clear articulation of biomarker rationale and model logic. Black box outputs without biological interpretation create skepticism. Integration between computational scientists and translational experts ensures interpretability and scientific credibility. The objective is not algorithmic dominance, it is probabilistic augmentation. As phase 2, failure rates decline, capital efficiency improves, even modest improvements in enrichment materially influence downstream portfolio value, reduced phase 3 attrition lowers overall capital burn. The narrative shifts from risk is unavoidable to risk can be engineered. This engineering depends on disciplined data integration, robust validation, structured biomarker strategy, and governance maturity. Organizations embedding machine learning into translational planning gain earlier clarity. Weak programs can be discontinued confidently. Strong programs can advance decisively. Emotional bias in decision-making decreases. Data-informed discipline increases. Artificial intelligence does not replace clinical judgment. It strengthens the informational foundation upon which judgment rests. The most effective leaders deploy AI selectively where heterogeneity risk is high and biological signal depth supports modeling. Digital biomarkers extend value beyond approval. Integrating them during development ensures continuity between clinical trials and real-world evidence. Regulatory dialogue strengthens long-term safety. Monitoring improves. Translational medicine represents the bridge between scientific hypothesis and patient impact. Machine learning, digital biomarkers, and predictive responder modeling increase the probability that this bridge holds. Phase 2 does not need to remain the most feared stage in development. With disciplined AI integration, it becomes a structured decision gateway rather than a probabilistic gamble. Leaders who act decisively will reduce attrition, strengthen mechanistic precision, and accelerate portfolio momentum. In an industry where incremental probability improvement creates exponential financial and therapeutic value. Phase 2 de-risking through AI becomes one of the most powerful strategic levers available. This is not experimentation. It is portfolio protection and pipeline acceleration engineered through computational insight.